Hello students, let us start with animal tissue. Starting with the animal tissue. Animal tissue. The animal tissue are broadly divided into the following categories. These are epithelial tissue, connective tissue, muscular tissue, and the nervous tissue. Today we are going to start with the connective tissue. The main function of the connective tissue, as the name implies, is to connect various parts of the body. Main function is to connect various parts of the body. This connective tissue is further classified into three categories, namely vascular connective tissue, skeletal connective tissue, and last is connective tissue proper. We will start our discussion with the skeletal connective tissue. Skeletal connective tissue. The skeletal connective tissue is further divided into two types, that is the bone and cartilage. Let us start our discussion with bone. The matrix of the bone is made up of both organic and inorganic component. The organic component of the bone is made up of protein, and the protein is called as ossin protein, whereas the inorganic component is made up of calcium and phosphate ions. It is made up of calcium and phosphate ions. It is the calcium and the phosphate which makes the bone rigid. Now coming on to the cell part, the matrix contains cell, and these cells are called as osteoblast cells. These cells are responsible for the formation of bone. It is responsible for the formation of bone. Now if we study the transverse section of the bone, say for example, this is the bone, and if we study the transverse section of it, then we will obtain such type of section. And if we study this section, in the center of it there is a presence of a canal. In the center there is a presence of canal, and this canal is called as the Haversian canal. There is a presence of canal and it is called as Haversian canal. This canal contains blood vessels. Here blood vessels are present. Haversian canal, it contains blood vessels. Now around this Haversian canal is the presence of, around the Haversian canal there is presence of matrix, and the matrix is present in the form of concentric rings. This is matrix, or this is arranged in a circular pattern. This is matrix, and it is arranged in a form of concentric rings. Concentric rings. And in this matrix are embedded cells. These cells are the osteoblast cells. These are what? The cells are embedded in the matrix, and the cells are called as osteoblast cells. These cells are responsible for the formation of blood vessels. Each of these osteoblasts have thin hair-like projections. These thin hair-like projections are called as canaliculi. Canaliculi. And the point of the matrix in where the bone cells are embedded is called as lacuna. Lacuna. Lacuna is what? It is the portion of the matrix, where bone cell is present in the matrix. It is a portion of the matrix where the bone cell is present is called as lacuna. Coming on to this canaliculi, these are small hair-like structures. These are small hair-like structures present on the bone cell. They help in the bone cells to communicate with the Haversian canal. This is the TS of the bone. The matrix in the concentric rings form. Center there is Haversian canal. Now there is one more thing that is called as the Volkmann's canal. Weitzman Canal. The Weitzman Canal is a canal which connects two Haversian canals. If this is a Haversian canal, the canal is going like this, and this is one more Haversian canal, and it is going like this. What it is containing? It is containing blood vessels here. Here also is blood vessel. These are the blood vessels. And what is the Volkmann canal? Like this. The canal which is connecting to Haversian canal is called as the Volkmann canal. So this is the Volkmann canal. So this is the characteristic structure of a mammalian bone. Now coming on to second type, that is the cartilage. Second type of connective tissue, that is the cartilage. Again coming on to the matrix. The matrix of the cartilage is just made up of the organic portion and that contains protein, and in case of cartilage the protein is called as chondrin protein, whereas the cells are called as chondrocytes. If we study the TS of the cartilage, what we will obtain is the presence of matrix. The matrix is not present in the form of concentric layer, it is just scattered, and in between there is presence of cartilaginous cells. These are the cartilaginous cells. So this is the matrix made up of protein, chondrin, and these are the cells. Now where is the cartilage present? What is the site where the cartilage is present? So it is present at the nose, ear, and forming rings on the trachea. The cartilage is devoid of any blood vessels. Now if we compare bone to cartilage, the first difference will be of the matrix. Here the matrix contain calcium and phosphate ions as well as protein oscin, whereas here only protein, chondrin is present, rest ions are absent. Second difference is presence of Haversian and Volkmann canal. Here no such canals are present. Third, matrix arranged in concentric rings, whereas matrix not arranged in rings. Fourth difference will be it is dense. it is comparatively loose. So this is all about bone and the cartilage. Coming on to next type of connective tissue, and the next type of connective tissue is vascular connective tissue. The vascular connective tissue is further divided into two subparts, namely blood and lymph. Coming on to blood, the matrix of the blood is made up of plasma, and which comprises of about 55% of total blood. It is present in liquid form. Whereas coming on to the cells, the three types of cells which are present in blood are RBCs, WBCs and platelets. Coming on to plasma, if we discuss about the composition of plasma, 90 to 92% of the plasma is made up of water, and remaining 7 to 8% is made up of consists of solid component. It can be either proteins, carbohydrate and various ions. Coming on to these cells, first is the RBC or the red blood corpuscles, also called as the erythrocytes. These erythrocytes are non-nucleated cells biconcave in shape or discoidal, like this. Main function is to transport oxygen. The lifespan of RBC is around 120 days, after which it is destroyed in spleen. This is all for the day. Thank you. In the next class, we will discuss about WBC.